work is in collaboration with Tom Givenish and Ricardo Kreipel. So what is my big picture goals? So again, it's to understand rhododendron characteristics and how that reflects pollinator-mediated selection and the environment in which they occur. So my approach towards this is really a macroecological approach. So I'd like you to think of it as if you're viewing an impressionist painting. It's best viewed if you step back and really look at the broad brushstrokes to try and understand the essence of what you're viewing. And in this case, we're trying to find the essence of what is a flower. So let's first back up a little bit and talk about expectations. What are some of the things that are predicted, at least based off of pollinator syndromes? And that has to do with the coevolution between flowers and their pollinators, and essentially how these kind of interactions shape the observed traits that you see. So in the instance of insect-pollinated flowers, the general expectation is that there can be a range of colors, as well as a wide range of fragrances. The flowers can be white, yellow, orange, this full array of different ones. Yet when you get to more bird-pollinated flowers, there tends to be kind of this transition instead towards predominantly red flowers that do not have scent. And part of that too is they also generally have a higher nectar content. And so there's a lot of ideas as to why that might be, but it may have to do with a combination of what kind of wavelengths of color can be perceived by different organisms, and also how can a plant essentially prevent less efficient pollinators from visiting and taking nectar, which is basically energy intensive for the plant to make. And so in the instance of red flowers that are scentless, that is going to be possible for a bird to see, but less likely for an insect. And again, C is and also the scent part of it. Again, detection. So again, these are some of the underlying expectations. So what does this mean in terms of the shift of traits along elevation gradients? And who are the expected pollinators? Well, it actually gets pretty complicated pretty quickly. Within temperate areas of the globe, you generally observe that you have more birds pollination at lower elevations. Yet in the tropics, it's the complete opposite pattern. You generally have bird pollination at higher elevations. So that leads to all sorts of questions as to why. And within that, is there something unique happening within the tropics for why there might be a very distinct trend? So obviously I was curious, does this apply to rhododendrons? And the short answer is, it looks like yes. So we have one of our speakers, Dr. Bassnett. Her research on rhododendrons of the Himalayas noted this trend of you have bird visitation that goes down as elevation increases, whereas bee visitation increases. Yet paradoxically, in the tropics, there's been a number of studies on rhododendrons there also, and they observed the exact opposite trend, that generally you have more like the characteristic of moth-pollinated flowers, which are white and fragrant, occurring strictly at lower elevations, whereas at high elevations, the montane areas, especially above 3,000 meters, were predominantly bird-pollinated. So that's pretty interesting. So again, is this a trend now that is specific to, just rhododendrons? Does it apply to a large geographic area? Well, there's another study that asks that question. This is looking at all the different islands across Southeast Asia. And they actually found kind of similar trends as the study in the Americas, in the sense of that lowland tropics tend to be inhabited by white flowers, whereas highland areas inhabited by red or pink flowers. And again, white generally indicating more insect pollinated and red indicating more bird pollinated. However, there is an added kind of complexity to this, in that there is also a shift in flower form that changed depending on the amount of seasonality, the amount of precipitation, and also getting into the shape and distance between islands. What is the degree of island isolation? And so that leads questions of, well, is there a biogeographic component also that shapes flower form? So this led to the following questions for me that I was like to ponder. And, first is, what are these pollination syndromes? Do they apply essentially to rhododendron? More specifically, what is the relationship between rhododendron flower color and scent? Next, how do rhododendron flower traits change along elevational gradients, and specifically within tropical ecosystems, so nearest the equator? And then the rhododendron clade within tropical areas is actually a fairly recent event. Um, but can they be shaped by biogeographic history when you have coevolution with pollinators that might have these kind of long-standing kind of barriers for their distribution? And then what is this kind of interplay again between pollinator-based selection and the location in which a plant occurs? And so thinking along about, again, along gradients, this kind of interaction of forces. And then lastly, what is known about pollinator types for rhododendrons? What are some of the major guilds that emerge? And then how does that relate to flower traits that are observed? And more importantly, how does that relate to kind of the differences between flowers that is observed? So now I'm going to stop and just talk about the data. So take a pause here. And this is the entire process of building a data set to ask these big questions. So first, I'm going to talk about the spatial distribution, the area of the globe that I'm interested in. Again, I'm interested in the island archipelago spanning from Borneo all the way to New Guinea. So that's referred to as the Malay archipelago, the Indo-Australian archipelago, Malaysia. There's a lot of different terms. Basically, I looked at herbarium records based off of all the different countries within this space to get an idea of essentially where do rhododendrons occur, their latitude, longitude, and elevation. And then I placed emphasis especially on species where there's pre-existing phylogenetic information. So that way we can begin asking more of these questions that is relevant to evolution, as well as accounting for relatedness. Next, I wanted to take a look at flower size metrics. So this information came predominantly from botanical monographs by George Argent. And this builds on, again, a huge amount of prior research and knowledge about the genus. And so that would be things like the corolla length and width, other things like the petal characteristics, the anther length, ovary length and such, but then also the ratios between these measurements to get an idea, again, of how does the shape change? However, for shape, I gotta say, Ricardo, he stepped it up a notch. uh, he helped develop these wonderful morphometric techniques. And so what you're looking at here are derived from photographs of rhododendrons from the front and from the side, and then using methods to basically quantify these very complex shapes. So rather than just the descriptive word of, oh, it's a trumpet shape or tubular, you now have a number to represent this. And this is work that built off of a prior paper written with Valerie Souza and investigation of bilateral symmetry, again, across these tropical species. And with that paper also came the phylogenetic knowledge that, again, I'm going to be working with. So additional metrics that I was interested in are things like flower angle. So essentially, are they erect, semi-erect, horizontal, hanging, semi-hanging? Also, the number of flowers within a display cluster. Information on are the flowers scented or unscented? Information on things like the relative abundance. So that would be the number of observations of herbarium records as an indicator of rarity. Details about flowering time, like how many months of the year do they flower and when. And then descriptions of color. And color, thinking of it as both a descriptive term, like the word white or red or yellow, but also as a quantitative way, as a continuous metric or the spectrum of color. And for this, I utilized, again, hundreds of photographs, sampling the RGB, basically hex codes for every single image. This is not as ideal as obviously taking a multispectral photograph that shows things like ultraviolet pathways on the flowers and patterns, but it's a lot better than just a few words to describe color. Now I have numbers. So below is basically the color palette of the species that I study. So approximately 100 different species, and again, sampling of their color. And in this instance, it is the primary color that is most commonly observed amongst the photographs sampled. So now, first part of the questions. Basically, is there evidence of correlated evolution between rhododendron flower color and scent? And for this, we're focusing on white or red flowers. So now let's take a look about the color distribution. And so this is best illustrated via a Venn diagram of all the different possible combinations of colors that a species can have. So in this instance, this is yet another way of quantifying color, where it's either yes or no for any particular color, and that does not exclude the potential for having multiple colors for a given species. So if you look at this, there are a few species that do have multiple colors, but they're relatively rare. The vast majority of the color spectrum is represented by either white or red flowers, and those flowers only have a single color morph. And so you can see that here, where you have 20 species that are purely white and 23 species that are purely red, but then all these various combinations within 5 different flower color descriptions. And now this is a step that I took where I basically read through hundreds of herbarium records and kind of reading the descriptions, either typed or handwritten, spanning over 100 years of knowledge. And this is mostly from the Royal Botanical Gardens in Edinburgh, but also utilizing digitized records from places like the Arnold Arboretum at Harvard. And one of the things about this that is important to note is that many of these field observations are not transcribed within the metadata. So that required actually, manually reading all these things. But you begin to notice a pattern that emerges where white flowers tend to have fragrance, and that fragrance may be described as things like jasmine, frangipani, soap, carnation. There's a range of different terms to describe that fragrance. Whereas red flowers are generally described as having no scent or unscented. Yet there are instances where you actually see the opposite, where red flowers may be scented, white flowers unscented. But from this, you can kind of get an idea of what is the frequency of distribution for these different color pairings. So now I'm actually going to take this and quantify it. And so for this is taking this information of binary traits and mapping it onto a phylogenetic tree and doing what is referred to as Pegel's test of correlated evolution. And from this, I want to stress the pre-existing prediction that we have coming into this is that there will be a shift from red scented flowers to unscented flowers will be the strongest kind of evolutionary direction as far as the changes in flower traits. And the results support that. So this is basically showing 2 different types of models. First being independent, that the 2 traits have nothing to do with each other, as compared to a model that is dependent, where there is a relationship between those traits. And then selecting the best model using the method referred to as AIC, Akaike Information Criterion, and essentially going for the lowest number. And if it's within 2 of the difference between the lowest, it could be either. So in this case, the best model shows that scent depends on color, but the next best model really suggests the opposite, that color depends on scent. So it's this kind of give and take that is suggested. Overall, however, there's you can get an idea of the directional shift via the thickness of the arrows that are illustrated on the model design. So here you have the strongest transition from red flowers that are scented to red flowers that are unscented as far as a character state. And to a lesser extent, you also have a shift from white flowers that are unscented to white flowers that are scented. So that supports our hypothesis. And overall, all these models, again, suggest that there is a strong connection between flower color and scent. And so that also, in turn, indicates greater kind of pollinator specialization. So now I'm going to think about how do these traits correspond with elevational gradients. So within the tropics, especially, there are very distinct vegetation zones as you go up a mountain. And so one of the obvious questions to me was essentially, are there pollinator zones that are comparable to these vegetation zones? And then thinking about the distribution of these different species of pollinators, what defines the uppermost elevational boundaries of their occurrence? Are there potential limits onto how far they can go based off of essentially thermoregulation or the metabolic costs of foraging? Like how expensive is it for them to fly, for instance? And that would be relative to things like external temperatures and kind of their body conditions. So this is a way of visualizing it. And this builds on this idea that essentially at low elevations, many types of organisms can exist. You can have a range of flower forms. But as you go up the mountain, it essentially becomes colder. You have greater differences between day and nighttime temperatures that may limit essentially which species can occur, and also thinking about, again, the costs of flying for an organism. So under this scenario, the prediction would be that nocturnal and especially hovering insects are going to do best in areas that are warmer, but they're going to be most limited by cold conditions. So you're going to have a filtering effect where essentially they cannot occur above a certain elevation threshold. Yet similar insects that instead forage at the daytime or that are heterotherms that have things like hair or shivering abilities or collective swarm abilities to maintain body temperature, they may be able to go slightly higher in elevation than other insects. And that at the highest, coldest temperatures, the montane conditions, that's where you're going to be able to find creatures that can generate their own body heat, and especially that can perch rather than hover as far as their foraging techniques. So again, thinking of this as the costs of flight and what are the expectations in terms of pollinator distribution and how would that change flower distribution. So again, low elevation, the expectation would be multiple flower colors, gradual transitions at mid elevations, and at high elevations being red bird pollinated only. That is the prediction. So let's take a look now at what are the distribution curves of richness for these different major kind of pollinator groups. So this is at the family level. This utilizes over 300,000 records, mostly of birds from places like eBird, and then taking their lat-long coordinates and interpolating it into elevation. And one of the important things when you view this figure is to understand that rhododendron richness peaks at around 2,000 meters. And by looking at this, you can quickly tell that everything below 2,000 meters, there's actually a wide variety of different pollinator types that exist. Their richness curves are very high within these lower elevation conditions. Yet, when you get to around 2,000 meters, there's a definite shift in how these distribution curves are, where you have a much higher probability of occurrence for bird diversity as compared to insect diversity. And partially this is also a reflection of what is the range size of the different organisms, where birds will have a larger range size than insects. That's based off of both the ability of how far they can fly, but also, again, potentially based off of body constraints and a variety of other distribution metrics. One of the other things to gather from this figure is that between the birds, honey eaters occur at the highest elevations, above 3,500 meters, where most other pollinator types are absent. Now let's take a look at the distribution curves of rhododendrons and their flower form. And in this instance, this is similar to the Venn diagram where a particular species can have more than one color as a potential. And from this, again, it reaffirms that the peak in richness for rhododendrons is around 2,000 meters. But also thinking about the lower and the upper elevational limits of those species. At lower elevations, essentially you have all the different colors of flowers are pretty similar to one another as far as the limits in which they occur. Whereas at the higher elevation areas, you begin having this greater differentiation based off of flower form. So essentially you can think of it as species that have yellow or orange flowers are going to be at a competitive disadvantage, and their abundance is going to taper off, their occurrence is going to taper off with elevation, as compared to red flowers that are going to be able to be slightly higher in elevation. Again, this is the uppermost elevation limits for where they can occur. And so now this is actually looking closer at some of the field observations that were done, especially in New Guinea. There is the observations again of red flowers at high elevations, white flowers at low elevations, but using it as more descriptive terms rather than quantitative terms. And this was done by Wade and McVean and Stevens. And so this is actually testing that idea. And so this is using a technique where, again, the flower color using these different metrics from the RGB code were basically simplified into a single color spectrum. And that's represented on the y-axis. And then on the x-axis is the maximum elevation which species can occur. There are two different slopes. The first slope is black. That is using regular linear regression. The red slope, however, is regression that is instead incorporates the phylogenetic tree, so accounts for relatedness between species. The main thing that you can gather from this is that yes, there is a significant shift in the flower color gradient from white tones to yellow to red as elevation increases. However, there's quite a bit of scatter. So it has an R squared, which is relatively low, 0.06. So that basically means that there's going to be a lot more to look at than just color. So now we're going to shift gears again and think even bigger. We're going to think about the biogeography of the entire archipelago and how that might influence, again, flower form. So we're going to back up a little bit and we're going to talk about animals. So this is an area of the globe that has a wonderful ton of islands. And it's a location that Alfred Wallace, in his expeditions throughout the archipelago, noticed incredibly distinct plant and animal communities, but especially animal communities, essentially at the intersection between Bali and Lombok, where essentially to the left on the Sunda side, that is closer to Asia, you have things like orangutans and tigers and all sorts of different birds, hornbill. Yet a very small geographic distance away, you have a completely different assemblage of animals. You have instead species that are more associated with Australia. And that leads to all sorts of questions. Why does that occur? Did it have to do with sea level? And only until later do we really have an understanding of things like plate tectonics and that the landscape as we see it today is not how it always was. So if you back it on up, like 50 million years or so, you'll notice that the landmass that is Australia and parts of New Guinea are actually positioned about 20 degrees south. And New Guinea as the island was mostly submerged. But over the course of tens of millions of years, this landmass began moving northwards. And so you had this entire assemblage of animals that were associated with Australia, especially montane areas, that went with it. Whereas the locations where Borneo is have basically been consistent along the equator this entire time. And so that led to a big biogeographic divide. And in the case of plants, that's also true, where you have families that are associated with areas of consistent equatorial position. You have areas in the middle Wallacea that have more of the recently emerged islands, whether that's changes of sea level or volcanic activity. And then you have areas like New Guinea or Sahul, which represents this landmass that moved upwards and northwards. And with that, you have different distribution patterns of endemism across different plant families. You have some families that are strongly affiliated with, let's say, just the Sunderland side. Those would be like diptecarps. Ericaceae, this is the family that rhododendrons are within. They actually have patterns of endemism across the entire archipelago. They don't have very broad distributions. They have very island-specific distributions. So now let's look at rhododendron and the biogeography and the history of this clade. So rhododendron essentially have temperate origins. They started off in the temperate foothills of the Himalayas. And they began expanding into the tropics. And keep in mind, this is the complete opposite pattern than what is mostly observed from most angiosperms. Most angiosperms start in the tropics and they go into the temperate areas. And recent analyses, especially getting into phylogenetics, have put the time span of this as rhododendrons spreading across the archipelago within the past 5 million years. In terms of time span, think of this as this is the clade is moving across the archipelago when the archipelago is in its current formation of the land masses. They're not going to be impacted by biogeography barriers in the same way that, let's say, a species that has been there for 50 million years will have that sense of evidence. So this leads to a very interesting question. If the rhododendron genus established fairly recently, are they going to be influenced by this longer, deeper time biogeographic divide? And the reasons why they might, it actually has to do with this coevolution of pollinator shapes, of pollinators influencing their flower shapes. And so this is going to especially influence vertebrates. And if so, then that may influence the flower size, curvature, or angle. And this is actually going to be less relevant for insect-pollinated flowers in that, in the case of things like hawkmoths, their ancestral state actually is a long proboscis, and they've been around since Pangea, meaning that they're not going to have quite the same biogeographic barriers in the same way that something like birds would have, based off of time. But to test this idea, essentially we took the rhododendron group and essentially divided them in half. And that most closely corresponded with Lydeker's line, which is similar to Wallace's line that I mentioned earlier in Alfred Wallace, except for that it's positioned closer to New Guinea or Sahul, so more eastwards. And then again, using these phylogenetic methods, referred to as phylogenetic generalized least squares. And so let's talk about different scenarios. Okay, so scenario one, it actually doesn't make any difference. And that could be because, again, rhododendron are fairly recent. It could also be a reflection of what is pollinating rhododendron, especially red flower type rhododendron that are pollinated by birds. In the sense of that in areas of the Himalayas and parts of Southeast China and other parts of Southeast Asia, you predominantly have more honey eaters that are the visitors. And honey eater distribution is very broad. It is across all of Southeast Asia and across the entire archipelago. So the prediction would be if they continue to be pollinated mostly by this group, then they're not going to show this kind of biogeographic divide. However, if there has been a shift to a new pollinator group that does have this kind of biogeographic divide, you might see a difference. So an example of that would be honey eaters, which have Australian origins. Their distribution is much more limited to essentially only the eastward side of the archipelago. Under that scenario, you would have a prediction that you might have differences of, for instance, again, flower curvature or angle. Even if you have convergent evolution between these different bird groups, the individual birds may have or species rather, may have different ways of accessing or foraging the flowers. They might have slightly different characteristics. Anyhow, let's see if that happens or not. So the results are, yes, essentially scentless flowers that are specific or throughout this area show differences based off of these biogeographic divides. Essentially, flowers within Sahul have greater curvature. They also have different ratios of the length width of the corolla. However, some of the other factors examined related to flower angle, corolla length, and a number of the previously mentioned metrics were non-significant. So what does this mean? Well, it definitely reinforces findings by Souza that noted a wider morphospace, but incorporating additional measurements again, but still reinforcing their findings. And then also field observations by Stevens that noted, again, the floras of New Guinea were very distinct, more flamboyant than the ones that were more westward. And one of the things that I find very interesting about this is that it's also a potential shift to honey eaters as bird pollinators of rhododendrons within this area. And one thing to think about, going back to the distribution curves I showed earlier, is that honey eaters really occur within some of the montane areas where other pollinators are absent. So potentially, even though you have these other types of birds like sunbirds, if they're limited to lower elevations, it means that as rhododendrons expanded into a new area, there's potentially an underutilized new ecological niche. And that niche was basically filled by honey eaters as being pollinators. So that's expressed in some of the flower form. Okay, so now this is getting at a very lovely example of citizen science. And anyhow, I'll just show you this. This is on BirdLife. I was very excited looking through all these photographs. This is basically showing a honey eater pollinating a rhododendron. So that's exciting. And look at the lovely curve of the flowers and how it matches the beak. So this is an example of a video that was taken that identified the bird, but did not identify the plant. And so that's, at least as far as future directions, an area that rhododendron enthusiasts and specialists can help begin linking some of this information as far as who's the pollinator. So now I'm going to begin thinking even bigger. And that in the sense of trying to create a model that incorporates all the previously mentioned concepts. So those would be concepts related to scent. Does that matter? Elevation, biogeography, and more importantly, all the different things that are important What is this interaction between them? What is the relative importance of, let's say, scent relative to elevation, which shapes flower shape more? And so for this is where we develop more complex models. So again, this is use of the phylogenetic generalized least square method, but it's also utilizing essentially lambda. This is the amount of correction to kind of the branch length that is determined by maximum likelihood. And our metrics are going to be scent as a yes, no, elevation, the minimum areas of occurrence, average, and maximum, but analyzed as separate models. And then bioregion, Asiatic versus Sahul, again, using mostly Lydeker's line as the defining area. And using a similar method as what I mentioned before with the scent to flower ratio, where the best model is determined by AIC. And if there are two models that exist that have similar numbers, like less than 2 difference, basically reading through that and deciding between those two models, which one is the simpler explanation, and opting for that as a way of explaining observed flower traits. And for this, it's usage, again, of the entire data set that I described below for all the different flower measurements, shape characteristics, color, all the above. One thing to note is that scent is used as a predictive, but it's more in the sense of thinking of it as scent as an indicator of pollinator specificity. So these are some of the top models, and top models that are defined by not only are they significant, but also is the R squared, or the amount of scatter, what is higher. So essentially, this reaffirms that color gradients shift, but more importantly is that they shift differently depending on whether or not the flower is scented or unscented. So essentially, if you have white flowers that are scented, they tend to be at lower elevations. But as you increase in elevations, there's a transition towards red flower tones. One of the things that also changes is flower angle, where scented flowers tend to tilt upwards, whereas scentless flowers that are mostly bird pollinated tend to tilt downwards. And again, this has to do with the angle in which a pollinator is most likely to access nectar for foraging. And so that as you go up in elevation also, the flower angle changes. It begins dropping down. There's also flower clustering, the number of flowers in a display, where if it is scented, essentially at lower elevations especially, you have very high flower clustering. So these very dense displays of flowers. Yet as you go up in elevation, the number of flowers decreases, and so you see more like 2 flowers per cluster. And that's a shift, again, also of how particular organisms may forage. So if you have a tight cluster for an insect, that's a very small distance to go. And for a bird that is very large, it may be better for there to be actually more space between the flowers for access. So that's why going for a lower density of flowers. And then taking a look at the side profile. And so this is using, again, the morphometric flower shape that has been quantified into a number gradient and simplified using a method called principal components analysis. And essentially with this, the petal flare begins to change, and where you have essentially at lower elevations, you could have more trumpet-shaped flowers. You have these deep lobing of the petals, and you have this great differentiation between the tube relative to where the petals are and what is displayed for the pollinator. But at higher elevations, you have this transition to more bell-shaped flowers. So that's part of the change. And again, also the petal lobing. So at low elevations, you have deeply lobed petals. At high elevations, you may have fused or overlapping petals. So let's visualize that, because again, this is what I previously talked about is, well, a lot of, of numbers in theory. So this is a way of visualizing it. So in this particular figure, every single point is a different species. The size of the point reflects the size of the flower. The color, again, represents that color sampling that I mentioned earlier, where changing the RGB into a hex code that can then be used for graphing like this. So the figure on the left, you can see the flowers, again, are more trumpet-shaped, deeply lobed type of petals. As you go up in elevation, you have this transition to more the kind of bell-shaped flowers with very minimal petals or overlapping petals. And this is reinforced whether you're looking at the side or the front. And again, red indicates, the red slope indicates the line from analyses that use the phylogenetic tree. The black line represents statistical methods that do not incorporate the phylogenetic tree. Okay, so let's stop again and take a look at a couple other metrics. In this case, it's the interplay between size and location. And basically the finding is that scented flowers tend to be pretty big, much larger than unscented flowers. But that is especially true on the island of New Guinea, or the bioregion referred to as Sahul, where regardless of the metric you're looking at, whether it's corolla length, tube length, anther length, things like that, everything gets bigger within that context. So this is an example of one of the largest, most flamboyant flowers, Rhododendron lycogygos. This is literally 10 times the size of the smallest flower within the same bioregion. It is a big old flower, size of my hand, maybe larger. So what does this look like, again, for some of these more complex models? Essentially, the side metric that encompasses curvature has an interaction between, or I guess is best reflected by, rather, whether the flower is scented or unscented and the bioregion. And that is exemplified, this difference, within specifically Sahul. So this figure shows kind of the curvature, where you have negative values represent curvature downwards, positive values is more curvature upwards. And the difference within the Sunderland Wallacea region is pretty minimal. Yet within the bioregion of Sahul, you essentially get curvier flowers. You get them going in all different directions. You go up, down, and then the differentiation again between insect pollinated and bird pollinated flowers becomes even more accentuated. So now let's talk about some of the pollinator knowns and unknowns and filling in. Okay, so we get an idea of the flowers, who's visiting them. So first, I gotta say, there are very, very, very few studies on pollinators within tropical areas, and that's obviously something that needs to be improved upon. But we can still take information that is gained from subtropical or temperate areas to try and fill in some of these gaps. And then utilizing additional information from observations from herbarium records and from photographs. So let's talk about more the generalist type of flowers, the best thing. These are flowers that are going to look closer to what rhododendrons look like within temperate areas. They generally are very colorful. There's a range of colors, very big petals, pretty large flowers. More importantly, there's kind of like a landing pad for how the petals angle outwards. And that provides essentially a location for multiple different types of visitors to visit, whether that's a bird or a bee or butterflies. And one of the characteristics of these flowers is that they generally have these very sticky pollen threads. And that makes it so that essentially pollination can occur via those entangling, let's say, the legs of a butterfly. And in the instance of, there's a study specifically on those threads, that tends to actually really help with pollination, especially with bird and Lepidoptera or butterfly-based pollination. So these are some of the examples. On the right, for instance, are actually two photos that I found just from going through miscellaneous photographs online, where people didn't necessarily identify what the plant was, but they took a picture of a bird or of a butterfly. So this is another application of where hopefully we can connect this range of information out there between citizens and scientists to try and identify, again, what are some of these relationships. And in the instance of hawkmoth pollination, this is a study that took place in southern China, Vietnam, where you have these white flowers. They're not necessarily the super I guess what you would expect as a hawkmoth pollinated flower, yet they're visited by hawkmoths. And more importantly, the study found that hawkmoths were much more efficient pollinators than bees were. And so that suggests that there may be a shift more towards hawkmoth pollination as you go towards the tropics. Again, they're more efficient. So that could be part of why you have particular shapes in the tropics. And I'm just going to actually just show you these shapes now. Okay, so this is a study on the left is looking at convergent evolution, basically similarity of flower form across many different families across the globe. And you notice these are all hawkmoth pollinated flowers. They all look like white trumpets. They're very fragrant. And this occurs in both the New World and the Old World. And guess what? On the right, you have rhododendrons that also kind of look like that. So it's not too much of a jump to imagine their chances are they're hawkmoth pollinated, especially when they have names like jasmine and florum and they have a fragrance like jasmine, which is hawkmoth pollinated. So anyhow, that's probably pollinating what flowers look like this. Next, we have bird flowers. And bird flowers are actually kind of interesting. These are some of the observations for the area that have been gleaned from things like the different herbarium records and notes about some of the species. There's been a formal study on rhododendrons in Mount Kinabalu and the pollination. And you can begin to see some of the relationships between flower curvature and beak type. Where sunbirds, you have these kind of dainty flowers, slight curve. You have a particular rhododendron, Malefagandum, hopefully I'm pronouncing that right, that is literally named after the same family as honeyeaters. And they have this very long, very pronounced curve. And then you have example like down below of the white eyes. They have a very short, relatively straight beak, and they're the primary pollinators of Rhododendron ericoides. That's a species that's specific to the mountaintops of like Mount Kinabalu in Borneo. And again, that is a species that occurs way above the tree line. It's like on exposed granite. So yes, birds can handle it up there. Okay, and then these are some of the surprise ones also, where you have, again, I mentioned these giant flowers in areas of New Guinea. Well, this is kind of like a pollination wildcard. Who visits them? And actually, a lot of studies have suggested, or not really studies, more like field observations, as well as kind of accounting from naturalists. So, like, there's a Birding Indonesia group about parrots visiting and pollinating rhododendrons. And one of the things that is definitely kind of complicates things as far as the assumption of bird flowers always being white and unscented. The thing is, lorikeets are one of the only birds that can smell. So that kind of disrupts that idea. But also, this is a group that when you read the accounts of what the floral fragrance is like, it sounds very distinct, or smells rather very distinct compared to other white flowers. And so that suggests that it actually might be bat pollinated, especially when you look at the flower shape. It has a much larger opening. That would suggest that the head of the organism that goes within the flower also has a larger or more blunt face shape. So that would make sense for bats or for, again, parrots. So again, the need in the future is to really fill in these unknowns, to identify who the pollinators are. So obviously more field surveys, but application of citizen science, it's also potentially screening these image libraries like what I have manually done, potentially utilizing AI. Also, some of the steps for the future would be doing more advanced spatial distribution modeling, taking the coordinates of these images for birds and of the particular plants and overlapping them. Which ones are most likely to co-occur? And then also incorporating measurements that were not incorporated within the study, and that would be analysis of things like the nectar sugar content or the chemical analysis of the scent, the fragrance that different flowers produce. And by the way, these are all images that I collected from Birds of the World, where there are different bird species on rhododendron, yet none of the photographs identified them as rhododendron. So again, let's try and fill in that knowledge gap. So now, what can we gather from all this? Let's tie this together now. So whether there are at least 4 different kind of flower types reflecting pollinator guilds. You have the generalist flowers that, again, are going to be more bee, butterfly, bird, other insects. They're going to be very brightly colored, larger, more open. They're going to also have landing pads for different organisms to land upon. And they're going to have especially sticky pollen. And then you begin shifting instead to these more specialized pollinator shapes. So you have the hawkmoth pollinated flowers for Sphingid, which are going to be strongly fragrant. They're going to look like little trumpets. They're generally white or pale, and they often have this kind of curved shape to them, more curved, kind of this downward to upward curve. There's also very high density of flowers, and these tend to be at lower elevations. Then you have the bird pollinated flowers, and those can actually be either curved or straight in the bell shape. But the main characteristic with them is that they're unscented and that they're usually hanging and in clusters more of like 2 to 3. And then you have these wild card kind of flowers, which are these extra big, giant, very fragrant flowers, but they're held more horizontally, which again suggests more either bat pollination or possibly parrot pollination. But those are ones that again are specific to only the island of New Guinea. So now let's visualize this in a kind of taking all the factors all at once, all the different measurements, so multivariate analysis, and simplifying it down to a two-dimensional form to think about what is the spectrum of flower forms that we observe. So this is referred to as a phylogenetic principal components analysis, and I'm going to walk you through how to read this. So essentially, you have these lines connecting the points. That reflects the relatedness between species. That's the phylogenetic tree essentially mapped on to these points. Every single point is a different species. Again, the size of the point reflects the size of the flower. The color, again, reflects the primary color that is observed for that species. And then whether it's square means unscented versus diamond scented. And again, this is put together using the flower measurements as described, all the shape metrics, color measurements, scent, angle, and flower number. And this explains essentially half the variance, or kind of why flowers look the way they do. So first, you want to read this left to right. So this is the referred to as the x-axis, and this axis represents the gradient of flowers ranging from kind of the spectrum of scented and unscented, from color and of size. So on the far left, you have Rhododendron ericoides, which is a red unscented flower, and it is very small. And on the far right, you have Rhododendron canori, which is an extremely large flower that is very scented and very white. Next, you want to read the y-axis, and this is what encompasses essentially flower shape, and where you have the positive part of the axis, the upper part, are going to be mostly symmetrical flowers that have more, again, radial symmetry, whereas as you go downwards to the negative values, that represents flowers that have a stronger curve, whether that is a red type of flower that's bird pollinated, like the Vireya in the lower left, or Gunnuffy on the right, that is more the hawkmoth. Again, you have this differentiation between more radial symmetry, simple kind of bell-like flowers or campulate flowers, to ones that are these curved, either trumpet or kind of these bird-shaped. And then lastly, in the center, you have what you can think of as just most the average, either that or things that are unknown. And this is the intersection essentially between scented and unscented. The distinguishing between them is kind of blurred. This is also where you generally have more yellow or orange flowers, or sometimes white flowers. And so you can think of the center part as being the space more of generalist flowers, whereas the outermost sections of this figure represent the more specialized flowers. And now we're going to take this and reapply it to how this changes with elevation. So this is using a metric referred to as functional dispersion, which is essentially how different the flowers are from one another within a given community. So you can think of it as the amount of scatter or spread. So if you think about the prior graph, it's like how big are all the points from one another? And essentially what this shows is that at low elevations, there's actually quite a bit of scatter. It means that the flowers are very distinct from one another. And most likely what that represents is that there's many different pollinator types within that area. However, at 3,000 meters, you'll notice a sharp drop in this metric. That means that the flowers are actually really all unifying towards more of a singular flower form. And that actually supports, again, this idea that you have bird pollination strictly at high elevation areas and this kind of filtering where other pollinator types simply cannot occur. So that, again, reaffirms our hypothesis and kind of ties us back to where we started. Now let's take a look at along a longitudinal gradient. So again, you have greater dispersion, greater differences between flower form, but on a longitudinal gradient, which is pretty mind-blowing. And if you think about this, it's as the rhododendron clade expanded across the archipelago, you have rapid speciation, and potentially you have many different new types of species with very, very different flowers by the time you get to New Guinea. So this also reinforces some of the other observations made by prior studies, but actually kind of quantifies it in a new way. And it gets at this other idea. Is it that via diversification of the kind of pollinators and the flower shapes that leads to speciation, or is it vice versa, kind of chicken or egg? Anyhow, I think it's pretty interesting. Okay, so now closing thoughts. We're going to kind of wrap this back up again. So again, understanding the relationship between flowers and their pollinators is an enigmatic pursuit. And the complexities are further entwined via the gradients in which the species occur, such as elevation, longitude, across a big archipelago. However, when you investigate these species traits and distributions at the macro scale, this kind of stepping back provides a unique perspective on this coevolutionary dance between flowers and their visitors. So again, step back from the painting, you can see the picture of the rhododendron. Okay, so special thanks. This study, I cannot stress enough how much this built off of such a massive amount of prior knowledge. I mean, again, 300,000 observations of birds went into this, hundreds of herbarium records. You have entire monographs of data that represent like hundreds of hours of people's study. Um, and then all the different research findings that help develop these ideas. So really, thank you. This is, you know, this study doesn't necessarily say anything new. It just shows it in a new way of quantifying it. But the amount of knowledge that this builds upon is something that I appreciate. And at this point, I really would appreciate any sort of feedback, questions, suggestions, or other ponderings, whether it's within this discussion session, or you can also just write to me, and we can talk about flowers. And then a whole lot of thanks to a lot of people. And also, lastly, in memory of people that helped inspire some of this research and provided their knowledge, but are no longer with us. So that's like George Argent, he wrote the botanical monographs, and Ben Hall, he was kind of this major figurehead for a lot of this phylogenetic work. And Anthony Lamb was really participated in a lot of the field studies within especially Sabah, Borneo. So in memory of them too. So thank you.
Thank you, Melissa, and we will open it up for questions. You can ask a question by putting your question in the Q&A. Oh, and you can ask your question by raising your hand, as Dylan has. Dylan, you want to ask your question?
Yeah, I have so many, but I'll stick with just one. Thank you so much for that amazing presentation.